Alright, hello. Today we're going to continue with our topic, which is protein quantification. So basically, quantitative proteomics attempts to simultaneously quantify level differences between main proteins and across different samples. So now, usually proteins are quantified with several methods, such as UV visualization, Lowry, BSA, BCA by chinchonic acid, and there are several other ways for you to quantify proteins. But however, it is not a measurement of the absolute concentration of the proteins. Some of these methods, all of these methods have their own drawbacks. So you need to know which method is suitable for your protein. It is important to evaluate effects and changes in proteins in all samples and test group, because sometimes phosphorylation also gives effect in quantitating your proteins. The primary indicator in deciding whether a protein should be investigated further. So also you need to pay attention to the indicator in deciding whether a protein should be investigated further or not. Probably not, or so. And also the differential levels of proteins can be reported as up or down regulated. As you know, when something is and when mRNA is transcribed, or probably we call that expressed. So there is a difference between upper regulated and down regulated. So in this graph, you can see that the number of proteins is different. If let's say the total sample is in the blue one, you can only identify the proteins, the certain proteins, only in the yellow ones. However, that means you will not be able to identify everything, because there is so much in your sample. And however, although you have identified most of your proteins, but still you will not be able to quantitate everything. Okay. In this overview of the current quantitative proteomics methods, you can see that there's this method called differential gel electrophoresis. So basically, the problem lies in when you have several types of gels. It is not easy for you to compare the expression, because there might be some other factors that contributes to the difference between expression from one gel to another. You basically, let's say you want to hold on. Okay, let's say you have two samples, sample 1 and 2, and you want to see the difference probably before and after of the expression. So you have and you tag them with internal standard called side-eye labeling. So which this labels basically fluoresces, and they are quite effective in binding with the proteins. Okay, so in this 2D gel electrophoresis, or DIGE, differential gel electrophoresis, your samples are quantified simultaneously. So, and also they are run together. That means because they have been labeled with the same with this side-eye label, it is easy for you to compare two sets of different proteins together. So it is much trustworthy than running gels at different times. When you have one gel that is run at the same time, it is easy for you to compare the before and after, because they have very it's coming from the same sample, but it has different types of treatment. Then you label them with different types of cyanide fluorescent dye. And then you run and you stain, and you can see and quantitate the protein that you have. So in this case, you have this part, which this one correlates to higher amount of expression of protein. Okay, and basically the image is read. The quantity is read with a side-eye specific scanner. Basically, you put it on the scanner, just like the scanner that you have at home, and they read based on the concentration using a special software. Okay, so like I said before, so this is the type of scanner that you use. You just put in your gel here, and you have the software that will read it out for you. And this is called the densitometer. Okay, the difference in protein levels quantified by measuring band spots or intensities is used. We use a densitometer. Okay, you basically put your gel in here. Let's say you cover your gel with a sarin wrap, or the plastics that you use in the kitchen. You cover it and you put it right here, and you close the lid, and the software will help to read it out for you. But however, the accuracy depends mainly on the choice of staining method. If let's say your staining is insufficient, so it will still be insufficient, and the type of software that you use, the shape of spot, and the background level. If let's say the noise is high, it will not be able to show as much as you would want it to be. So in this case, fluorescent dyes are the most popular since signal intensity corresponds accurately to the number of protein copies in the spot or band. Okay, most of the time we always use fluorescent dyes. Okay, it's really precious. So in this figure, if let's say you have your gel, and the densitometer came out as a figure like this, and they help you to calculate each of the bands that appear in your in the figure that is taken that you took. Okay, so imaging system, you have several types right here, and you can go online and find the software and imaging system, and basically they help you to compare and tell you and help quantitate the amount of protein you have. Okay, I'm gonna show you how it looks like, the differential. Before we stop, I'm gonna pause for a while to show you the differential proteomics. Okay.
Stable isotope labeling with amino acids in cell culture, or SILAC, is a simple and accurate method to quantify differential changes in the cellular proteome. SILAC uses metabolic incorporation of stable, non-radioactive deuterium, carbon-13, or nitrogen-15 labeled amino acids into proteins using specially formulated media. Typical experiments involve growing two cell populations in cell culture media that are identical except that one contains the natural amino acid referred to as light, and the other contains the isotope labeled amino acid referred to as heavy. After cell lysis, equal amounts of protein from both cell populations are combined, enzymatically digested, and analyzed using mass spectrometry. SILAC amino acids are chemically identical to natural amino acids but differ in mass due to the incorporation of stable isotopes, denoted here by the red asterisks. These heavy amino acids, typically lysine or arginine, facilitate detection and quantitation of labeled proteins' relative abundance in different samples. Up to 4 different cell populations can be analyzed using thermoscientific new code amino acids. These heavy amino acids contain different stable isotope variants that can be analyzed using high-resolution mass spectrometry. Unlike traditional SILAC that compares light and heavy samples, SILAC with new code amino acids is used to compare multiple heavy samples in the same mass spec analysis. In An example of a new coamino acid is lysine 202, which contains 2 heavy carbon atoms and 2 heavy nitrogen atoms. This amino acid can be paired with lysine 040, which contains 4 heavy deuterium atoms. Since both amino acids have the same nominal mass, they are indistinguishable at low mass resolutions, but are resolved using high-resolution mass spectrometry for quantification. Here we describe the steps to metabolically incorporate heavy amino acids into cells prior to protein digestion. All reagent preparation and cell culture must be done in a tissue culture hood under sterile conditions to prevent contamination of your cultured cells. The first step is to prepare the amino acids by dissolving 50 milligrams of each heavy or light amino acid in 1 milliliter of medium and mixing thoroughly. The color of the medium will change from red to yellow upon addition of the amino acid hydrochloride salts. However, the color of the phenol red pH indicator will revert back to red after addition to the rest of the medium. The next step is to prepare the medium for the addition of the amino acids. Remove 50 milliliters of the silac medium from each bottle and replace it with 50 milliliters of thawed dialyzed FBS. As silac medium is deficient in both arginine and lysine, both amino acids must be added to the medium to support cell growth. For experiments using only lysine for isotopic labeling and subsequent enzymatic digestion, supplement each bottle by adding 50 milligrams of the dissolved light arginine. Next, add 50 milligrams of dissolved light lysine to one bottle and 50 milligrams of dissolved heavy lysine to the other. For experiments using both lysine and arginine for isotopic labeling and subsequent tryptic enzymatic digestion, add the light lysine and arginine to one bottle and the heavy lysine and arginine to the other. Once the amino acid solutions have been added to each 50 milliliter bottle of medium containing dialyzed FBS, mix thoroughly. If required for a specific cell line, include additional antibiotics and media supplements before mixing. Sterile filter media containing dissolved amino acids using a 0.22 micron filter flask into bottles labeled heavy and light. Medium that has been supplemented with serum is typically stable for 3 to 6 months. Always store the medium at 4 degrees Celsius protected from light when not in use. The next step is to prepare your cell lines for incorporation of the isotope labeled amino acids. Depending on the cell type, split between 1 and 200,000 cells adapted to grow in the specific medium type into 2 or more tissue culture flasks or plates, with one containing the heavy medium and one containing the light medium. For suspension cells grown in T25 flasks, use 8 milliliters of each medium. For adherent cells grown in 60 by 15 millimeter plates, use 4 milliliters of each medium. Passage each cell population for at least 5 doublings by changing the medium or splitting the cultures as appropriate every 2 or 3 days. Maintain the density so that the cells are actively growing in log phase at 30 to 90% confluency. After 5 cell doublings, the incorporation of heavy amino acids should be greater than 95%. Harvest 1 million cells from each labeled cell population to determine isotope incorporation efficiency. Prior to mass spec analysis, the cell should be washed twice using 5 cell pellet volumes of PBS and centrifuge for 5 to 10 minutes at 500 times G to pellet the cells and remove the silac medium. The efficiency of isotope incorporation is determined by liquid chromatography mass spectrometry analysis of the samples labeled with heavy, silac, or new code amino acids. Standard in-solution or in-gel sample preparation techniques can be used to extract, reduce, and alkylate cellular proteins before enzymatic digestion with lyse protease or trypsin protease to generate peptides. Peptide samples are then analyzed by LC-MS to determine the percent incorporation of heavy amino acids into the metabolically labeled proteins. The percent incorporation is determined by assessing the peak area of light and heavy peptides observed in the LCMS analysis. This is facilitated by database searching of tandem MS peptide spectra using the heavy amino acid masses as dynamic or variable modifications. Once full isotope incorporation has been determined, continue to expand the light and heavy amino acid labeled cells to the number required for subsequent cell treatment and lysis. To simplify your SciLac experiments, we offer complete kits for lysine or trypsin digestion. Each kit contains SciLac media, dialyzed FBS, light and heavy amino acids. Individual components and other media types, including SciLac DMEM, RPMI 1640, DMEM F12, MEM, and IMDM, can also be purchased separately. We also offer multiple pack sizes of heavy and light lysine, arginine, and leucine amino acids. Nucleotide amino acids are our most recent breakthrough in SciLac quantitation, enabling multiplex quantitation up to 4 samples in a single LCMS analysis. Find out more about our SciLac metabolic labeling systems at thermofisher.com/SciLac.
Alright, so proteomics analysis of these adaptations to environment. If let's say you have 2 samples, one is ethanol with ethanol limitation and the other one is with glucose limitation. It's about the same difference. You can see that in every protein it has a difference in expression of protein. So they compare and helps you to read out the quantitative protein, and also a comparative protein analysis of macrophages. So probably you can spot the difference that it has. It looks similar, but it also looks different. Okay, and it has a different cyanide label, Cy3, Cy5. Next one we have MS-based quantification, which this one is very dependable these days. It utilizes internal standards. So just like BSA, if let's say the ones that you've done in the lab, you use BSA as your standard. You make a standard before you start reading your protein. So basically, internal standard is something that you put together, okay, to put together with your protein, and usually they are compounds that have similar properties to the analytes and added to the samples. Okay, most commonly used are stable heavy isotopes for samples and light isotopes for controls. Okay, so you have to note this. It can be added to samples at variable stages, in vivo or in vitro, and basically you can follow all this. We have SciLac, stable isotope labeling amino acid in cell culture. So they add this isotopes in the cell culture to read out as tagging. Okay, if let's say you're doing metabolic labeling, you can mix the cells in the beginning, and in the end you can read out through MS with the mass to charge ratio. But for protein labeling, this one is bigger. Mix of proteins, and you can read out the charge. And for peptides, they are basically small. You can mix them in at the final, and they came out as same thing, mass to charge ratio in MS. This one is much easier to read. Okay, the principle of SciLac. Okay, if you don't remember, SciLac is stable isotope labeling amino acid in cell culture. So let's say you have this one is using light isotope. This one is with heavy isotope. One is the control, the other one is the testing and with the treatment. Okay, this one is control, this one is the one with the treatment. You harvest and lyse cell, quantitate, and if you can excise this band let's say this is the event of interest, and you can do trypsin digestion and read out the protein abundance. Okay, so the abundance is basically depicted by the intensity. The stronger the band, okay, the stronger the band, that means it has a higher amount of protein. Another example is this is the control without treatment. This is with the treatment. And so you compare them, you can see the abundance. You compare between light and heavy. Okay, so in this case, the peptide has both the same abundance in both samples. And another one. Okay, so this one is like the control. This one is the one that is treated. Okay, the types of in vitro labeling. You have proteolytic labeling, incorporation of oxygen isotopes, and you have ICAT, isotope coded affinity tag, with by using all these reagents. And the advantages is high specificity, high sensitivity, applicable to most samples for ICAT. So comparison between SILAC and ICAT. So the difference in this one is cell culture with normal arginine. This one is with heavy arginine. And this one you have a light linker and a heavy linker. And in the end, okay, that's from the light. This is from the heavy. Well, the same thing. Okay. You can take some time to help you out in understanding this figure right here. But basically, I would say this one is much easier because you're using purified protein, so it is much specific as compared to SILAC. Okay. Next one is iTrack, isobaric. This one is by using pressure. Everything you can put up to 8 samples. You have lots of samples, but they are given with the same pressure. A moiety reactive towards amino groups, a balanced group, and a reported group. So these are the 3 components in iTrack. So like I said, iTrack has more samples, but they are using more. They're using the same pressure as a comparison by maintaining volume. So and then they read out the identification from peptide backbone. Okay. Okay, so one of the examples of a research that is conducted using iTrack is an allograft rejection by identifying differentially expressed hair proteins at defined stages. So they are looking at different times, like this one. So this has 0, 30, 60, 120 minutes. So they have several types of things to be compared, and they will combine, quantify, and identify. Okay, such as this one. Several samples of about the same thing. Combine and quantify and identify at the same time using MS. So this is the method. And for example, right eye allograft. Then you pull them and you will identify and quantify right here. Figure 3. Let's say you have a different period, 2nd day, 7th day, 14th day, and you pull them together and you put it into MS and you read them out. Alright, thank you.